Transcription is the process by which genetic information in DNA is copied into RNA, which can either act as the template for protein synthesis or perform regulatory or structural functions. In prokaryotes, transcription is relatively simple and closely linked to translation, reflecting the absence of a nucleus and limited RNA processing. Prokaryotic transcription is carried out by a single RNA polymerase holoenzyme composed of a core enzyme and a sigma factor. The sigma factor provides promoter specificity by recognizing conserved sequences, notably the -35 region and the -10 Pribnow box. Binding of RNA polymerase to the promoter forms a closed complex which unwinds locally to generate an open complex, allowing RNA synthesis to initiate. RNA polymerase reads the DNA template strand in the 3' to 5' direction and synthesizes RNA in the 5' to 3' direction using complementary base pairing with the uracil replacing thymine. After a short RNA transcript is produced, promoter clearance occurs, and RNA polymerase enters the elongation phase. Elongation continues until transcription is terminated, either by rho-independent mechanisms involving RNA hairpins followed by a poly-U sequence, or by rho-dependent mechanisms requiring the rho helicase. Because transcription and translation occur in the same compartment, ribosomes can begin translating mRNA while it is still being synthesized. Gene expression in prokaryotes is therefore primarily regulated at the transcriptional level. A common regulatory strategy involves operons, in which functionally related genes are clustered under the control of a single promoter and transcribed as a polycistronic mRNA, enabling coordinated expression. Regulation of operons may occur through repressors or activators that respond to environmental signals. However, not all operons are regulated in this way, as some are expressed constitutively or are controlled mainly by promoter strength or sigma factor availability. Additionally, many prokaryotic genes are not part of operons and are regulated individually by similar transcriptional mechanisms. Regulatory proteins bind to operator or promoter proximal regions to modulate transcription.